450,000 cases a year of heart failure in the US, and yet only about 4,000 or 5,000 heart transplants.
Dr. Mark Skylar-Scott is an innovative engineer and researcher at the forefront of 3D bioprinting technology.
There's good reason for optimism, and there's good reason for the forward motion we're seeing in this space of organ manufacturing right now.
His groundbreaking work is transforming tissue engineering and regenerative medicine by creating functional human tissues and whole organs.
What if we could engineer the cells to grow faster, or to not need the expensive ingredients, to make biology much more scalable?
In our conversation, Dr. Skylar-Scott shares his journey from the lab to the clinic, the challenges he faces and the breakthroughs that are paving the way for a future where organ shortages may be a thing of the past. There might one day be a heart that could be made from your own cells. Join us as we explore the incredible potential of his work and its impact on healthcare. Welcome to Stanford Department of Medicine's Inside Look at the Future of Medicine. I'm really excited to spend a few minutes talking to you about this incredible work that you've been doing. The idea of printing human organs, and in particular printing a human heart, is revolutionary. It's also a very big, bold idea. When did this idea come to you? It's a little bit crazy.
Yeah, I'm glad you described it as bold and crazy, because it is bold. It's really, really hard. So from the outset, I think it's really important that people know that organ manufacturing is a big and hard problem.
Yeah.
It is going to take decades. It is going to take a lot of dedicated hard work and a lot of new technologies to make it real. But there's good reason for optimism, and there's good reason for the forward motion we're seeing, in this space of organ manufacturing right now. So in terms of when I sort of had the idea to make organs, it's been a long process of evolution in my own mind.
Yeah.
I enjoy building things. I've always enjoyed building with biology, trying to manufacture pieces of tissue. And I think it was really sometime around early 2010, 2012, at that sort of time, when we started to be able to make blood vessels, that I thought, blood vessels is something every tissue needs.
Yeah.
And without them, you can't make anything large, you can't keep anything alive.
Right.
So with the ability to make blood vessels, I started to just think bigger. Why don't we make, a thick piece of tissue? And from there, why don't we make even bigger bits of tissue? And I got really inspired by the need for organs, right? It's a very large societal problem.
Yeah.
450,000 cases a year of heart failure in the US, and yet only about 4,000 or 5,000 heart transplants.
Right.
So the vast majority of people that succumb to heart failure don't get access to an organ, and we want to change that. So it's a really big, really hard problem, and I think that as an engineer, that's a really fun space to work in.
Yeah, incredible. I mean, the fact that there is such a huge need, and the fact that we spend so much time dealing with the consequences of implanting organs that are never going to be perfectly immune matched. Well, that's something we look forward to getting into in a minute or two. I'm fascinated by people's backgrounds, and you alluded a little bit to how you always like to build things. So just take me back all the way.
All the way.
Did you know from birth that you were going to be an engineer? What was your childhood?
My dad's an engineer. My brothers are engineers. My granddad's an engineer. So I think engineering was always kind of in our family. My dad was a mechanical engineer and worked in the metallurgy. I'm from Sheffield in England, which is kind of the steel city.
Yes.
And I remember him distinctly sitting me
Cutlery.
down and saying,
Silverware.
"Cutlery, you make very good knives."
Yes.
I remember him sitting me down and saying, Mark, it's not a great time for the steel industry in Britain. If you do want to do engineering, do something that's, the sun is rising on, not where the sun is setting on. So, I think that probably shaped some of my thinking to go into bioengineering.
Yeah.
But that didn't exist in the UK at the time I was an undergrad. So I did general engineering, and my course really described the need as an engineer to learn not just your subspecialty, but really understand the breadth of engineering. So my course kind of prided itself that in the same exam where you need to understand soil mechanics and how structures fail, they could ask questions on power lines or biological engineering. They really described this kind of integrated engineering, and so it taught you to pick bits of information from lots of different places. And I think that was very helpful for me to get a breadth of understanding of how to analyze complex systems.
Yeah.
It wasn't until I did my PhD where I
Yeah.
could come to the US and start my bioengineering degree at MIT and Harvard.
And how did that happen? I mean, that's a big leap across the. I did
Yeah.
a similar leap myself.
Yeah.
I lived a little
Depends
further.
who's asking. I mean, the
Yeah.
honest answer is
Yeah.
love.
Yeah.
I actually did 1 year as an undergrad at.
We love honesty on the future of medicine.
Yeah,
here.
yeah. So I met my now wife as an undergrad and got back to the US to be with her. But
And
also
and you met her in the UK
at MIT on an exchange program.
see.
So 1 year of my undergrad was at
Yeah.
MIT, and
I see.
that sort of brought me into the amazing world of American academia and also my now wife. So I jumped across the pond and started a degree in bioengineering, which is where I'd always been
Wanted
interested
to be.
in doing my.
And in the UK, you were in Cambridge.
Cambridge,
Yes,
yep.
right.
So my master's degree at Cambridge was looking at actually the energy efficiency of neuron transmission in the brain.
Okay.
And that was a very theoretical sort of computational exercise, and I always wanted to be more experimental. So that's when I came to the US, I took the chance not to understand how the nervous system is wired, but to actually control how it wires.
Yeah.
And so I built my first 3D printer in my PhD work, which used a laser to scan and try and direct how neurons would grow their axons and dendrites to create little networks.
Wow, interesting.
And I started to try and make those bigger. And what happens when you have lots of neurons in 3D tissue?
Yeah.
They died because they couldn't get vasculature. So that was kind of the realization that, wait, we can use these printers to make little tiny channels and feed our thicker tissues. And I think that's when I really got hooked on tissue and organ manufacturing.
Yeah, that's really interesting. I mean, one of the things, first of all, on your original topic, of course, it's an important sort of part of our understanding now of AI, and the idea of like the efficiency of the human brain is really one of the unique elements where we're learning. It would seem as AI advances further and further that there maybe aren't as many unique elements as we thought.
Yep.
But still, large-scale computing uses megawatts and more.
And yet we need 2 hamburgers a day, right? It's kind of
our brains are pretty amazing.
It's pretty amazing. The energy efficiency of the brain is mind-blowing.
Yeah. But I love this idea that it was really the idea of you were trying to scale up tissue and sort of came to this idea that you just reach a limit as to where oxygen can diffuse, essentially. And that's why, much in the same way that evolution, I guess, has provided us with blood vessels.
We have the solution inside us, right? Everywhere, yeah.
Yeah. So this all, the seed of this all started with your PhD.
Yeah. So, towards the end of the PhD, I had this very nice laser printer that could write beautiful, detailed, tiny little capillaries, but everything was the size of a pinhead. Right. So I kind of saw the potential for vascularization, but was disappointed in the scale at which you could write with these little laser systems. And so I really wanted to begin scaling that up. And actually, when I finished my PhD, I was initially planning on starting a company with a couple of friends, but that actually fell through right at the very last moment as I was defending my thesis.
Really?
So I had quite a quick pivot to work at a different startup called Formlabs, and that was a 3D printing company that's now quite large. There were maybe only 10 of us when I joined, and now it's about 1,000 employees. So I got to kind of experience the startup world of a really successful startup.
And so I think most people have some sense of what a 3D printer is, but maybe just for those of us who don't spend our everyday thinking about 3D printing, just maybe explain a little bit about what that is.
Yeah, there's loads of different flavors of 3D printing. So obviously 3D printing, you want to make a 3D object, right? So you need to define shape where you want bits of material and what material where and where you don't want material. And there are 2 broad classes of 3D printers. One is called extrusion 3D printing, where you squirt stuff out of a nozzle, right? You move a nozzle around and in 3D, squirting cells or material, plastics, building up a 3D shape. And the other approach is light-based 3D printing, where you project a pattern onto a vat of material or resin that turns from liquid to solid when you expose it to light. And so then by projecting layer by layer, different patterns, you build up a 3D shape. So those are the 2 broad classes of 3D printing. There are many in industry and homes, right, that you're used to seeing with printing plastic, and bioprinting remains still on the research end, but bioprinting is just printing with a 3D printer that is designed to print with biology, to print cells, but is very similarly, designed to a normal 3D printer.
Yeah. Do you know who first had that idea? Like watching plastic extrude out, or they said, oh, I could do that with cells.
Yeah, there's actually, for such a big industry and such a big story with a lot of technologies, a very clean answer to that. So Chuck Hull published the first patent in 3D printing in 1984. And for some of your listeners, that's quite old.
Yeah.
I think most of us heard of 3D printing in mid-2000s.
Absolutely, right.
And yet this is a technology that really came out in 1984, and there were a few big behemoth companies selling these $500,000 machines to the big industry partners. But what do you know about patents? How long do they last?
Yeah. Well,
20 years.
20 years,
right?
right.
So 1984 plus 20 is 2004.
Yeah.
So the reason why it really entered the public imagination in the mid-2000s is because the patents started expiring, and all these new players in innovation were just flooded into the field, and printers appeared everywhere, and are now, for any engineer, just a standard part of our practice in life.
And I notice even on the social media channels on YouTube, there are entire channels of people like devoted to 3D printing useful things for around the home, or and people with whole almost like factories of 3D printers.
Yeah, I think they're an incredible tool for engineers and hobbyists alike. I think maybe in the height of the bubble, people were thinking everyone's going to have a 3D printer, right? I don't think that's come to pass, but I think that actually hides the true revolution, which is in engineering labs and designer labs, where printing is now absolutely essential for anything in these fields. So in terms of bioprinting, there's also a single person that is usually credited with the foundation of bioprinting. That's Tom Boland. Professor Tom Boland took an HP printer, so an inkjet printer.
Like an inkjet?
Literally an HP inkjet
amazing.
Printer.
Yeah.
And instead of connecting it to a vial of ink, he basically plumbed in cells and started pushing cells through. And that created the first sort of inkjet deposition of cells. And
that's amazing.
That kind of got the field inspired into manufacturing.
What did he print them onto?
Just onto a glass substrate. So it was quite thin. Because the thing with inkjet printers is it needs to be a very low viscosity material, very watery. So when it hits that material, it spreads. So it's hard to get a good 3D structure with that process.
And I guess you learned at that point that some cells would survive that process and some might not, or are they pretty robust?
It's pretty robust. So because you've got low viscosities, the forces involved are quite low. You don't need much force to push a low viscosity material. So the viability is generally pretty good for a well-designed printing system.
Yeah. So you're at this startup company.
Yes.
And you're learning all about printing.
And Formlabs was awesome. It was great to be there at a time where I could experience like explosive growth, right? They had the biggest Kickstarter at the time in history. And I got to really witness a company develop and a culture develop. But I really didn't want to print plastic for life. I really wanted to get back into the bioprinting world. So after a short stay, I only stayed 6 months. I then was able to find a postdoc at Harvard with Dr. Jennifer Lewis. And she had just moved from Illinois, so it was perfect timing. And she wanted to start this bioprinting program, and her technology that uses extrusion printing, unlike those little pinhead tissues with a laser, this really was the way to make big, viable tissue. So it was a very obvious decision for me to get into that environment.
At that point, did you have specific ideas of where you wanted to go? Did specific organs?
No. I just wanted to go big and thick, right? Every organ needs vasculature. So I was in a lucky position of not really needing to choose an organ. So for me, it was, look, can I just keep a thick centimeter wedge of tissue vascularized and living? That's a pretty low bar, but I would argue a
very
really important one. Because if you can't keep your tissue living, you can't do any of the next steps. So we actually began with mesenchymal stem cell printing. We made a centimeter of mesenchymal stem cells. These are cells that form a lot of the connective tissues in your body. And we could then flow instructions through the printed blood vessels to tell those stem cells to become bone-like cells. So that was sort of the beginning of that thick tissue work.
I didn't realize, I've been more familiar with the work that we'll come to that you're working on now, and specifically focused on the human heart. but I think I hadn't realized until I was reading a little more about your background and as we're talking here, just how that you're thinking about this, about how integrated the vasculature and the tissue needs to be. That was really your starting point in many ways. I think a lot of people start with the idea of an organ and presumably reach a stop at the point they realize that if they don't solve vascularization, they're at a stopping point. But from your original thinking and ideas around this, you already were thinking about the biggest challenge being the vascularization one.
That's right.
Yeah.
And it's also the one that 3D printing is best addressed to manage, right? It's you've seen these casts maybe of blood vessels in the past. It's a very complicated 3D structure, the blood vessel tree. And that structure is actually important. You can't make it like a lattice or a climbing frame sort of structure. So it has to be
maybe say a few
truly
more
a tree.
Words about that, because I think a lot of people really do think about blood vessels as, hoses down which blood flows.
Yeah.
But of course they are their own complex organ. So maybe say a little bit about that biology of the blood vessel, but also how that biology creates challenges for you.
Yeah, I mean, perhaps a good starting point is a simple analogy like driving across the US, right? If you want to create a US postal system and that postal system had to run, just on small roads, it would take a long time to get your mail from east to west coast, right? We have highways to handle fast-moving, long-distance traffic. At the same time, if all we built were highways, right, you wouldn't be able to deliver.
No towns.
There'd be no towns. We'd all be 90% by volume, by area road. So you need this combination of large arterial, we even call them arterial roads, right? You need large arterial roads and then smaller side roads for the actual houses and delivery. And it's exactly the same thing with the vascular tree. If you are designing a small piece of tissue, just like looking at a carved out piece of suburbia, probably doesn't matter. You can get away with all small roads, capillaries, the smallest vessels. But when you want to do multiple scales, big heart, smaller tissue, right down to the cell level, across all those length scales, you need a tree to distribute across the body, across the organ, all of that blood and oxygen, and then deliver efficiently through the small vessels. So biology figured it out, right? They have the vascular tree. And the tree is not just structurally complex, but biologically complex. So these big arteries, right, they have a different structure to the capillary. The big artery is designed, evolved to have sort of pressure handling capacity. So it has an inner lining of blood vessel cells, these endothelial cells that tell the blood everything's fine, don't clot, be happy. Right? And behind that you have smooth muscle cells that gives stiffness and elasticity to help the vessel withstand pressure. They also allow the vessels to respond to hypoxia. So if a region of the tissue doesn't have enough blood, it can sense that and actually relax those muscle cells and redistribute flow, which is kind of unbelievable. I'd love us to have engineering solutions to that without biology, but that's even distribution across these networks is very difficult without that feedback from the smooth muscle cells.
Just on that point, we do, a group of patients come to mind who end up without that elasticity in their arteries. They have a cardiomyopathy, a heart muscle disease. They'll often complain when they get up and start walking that they have chest pain because that elasticity isn't there. And over time, 10 minutes later, those blood vessels have slowly opened up because of sort of circulating bio agents. But at the time they want to move, their arteries don't move as quickly. Now, I think most of us don't realize that our body does this automatically. The minute you start to think, I'm going to exercise, your body's already working on those arterioles.
Or stand up from your chair, right? As a tall person, it's a real issue when you raise your head. You can get a little lightheaded for a couple of seconds, right? And the muscles constrict, ready to prepare, the body for that gravitational effect of blood pooling.
We take it for granted until it's not there.
We do.
But just one part of the tree, right?
And that's where I think engineering meets biology is important, because engineering the complexity of the human body is really, really hard. We can't necessarily design our printers to print everything and have all of those functions. We rely on the cells, right? Cells are intelligent, right? They have in their DNA the information programmed into them on what they should be, where they should be, and how they should function. And we need to rely on that because right? You've seen our humanity's efforts to make humanoid robots, right?
Yeah.
It's moving along pretty impressively, but
Right.
it's still far behind the amazing dexterity of the human body.
Yeah, yeah. Well, you were talking about the arteries and the arterioles that were built for pressure. Capillaries as well, have a very specialized function for exchange of nutrients and oxygen.
That's right.
How do you think about
Yeah, so you need
designing
a very high surface area, right? You probably heard in a similar kind of way, the surface area of your lungs, if you were to open them out, is about a tennis court. I think that's always the textbook example. That's extremely hard to print, right? The blood vessels in your body are tens of thousands of miles long, very, very long, amount of complexity. We cannot, there is no technology out there yet that is able to print every capillary at the resolution we need and every big vessel at the scale of an organ. That doesn't exist.
Right.
So we rely on the cells creating their own capillaries to have sort of functional networks. And luckily enough, given the right conditions, blood vessel cells will naturally form capillaries.
Form tubules.
Form tubules.
Wow.
Yeah, it's kind of amazing. Yeah. But I mean, of course, we self-assembled ourselves, right?
Right.
That's
but we're not.
life.
We're always not. We have such an artificial state in our science labs. We're not necessarily used to being able to
give the cells the
recapitulate.
correct milieu, right? So endothelial cells, right? These are the lining cells of the blood vessels. Give them the right support cells like skin cells, fibroblasts, macrophages, and they will self-assemble into a tubular network. It's called vasculogenesis, and I mean, it's very handy. It means we don't need to print maybe every single capillary. So that's, I think, going to be the key, where we as engineers don't tell biology everything. We facilitate biology. We don't print all of it. We have to give it kind of a template and a big structure, but let biology take over at the small scale.
How much of that is dependent on our ability to sort of push the cells back to a more embryonic form or a stem cell-like form?
Certainly for the cell manufacturing, stemness is very useful. So a stem cell has two jobs, divide and recreate itself, right? Or to differentiate to create a cell type of interest. And when we think about organ manufacturing, actually almost even before printing the blood vessels, right, you got to get the cells. And the cells for a solid organ, we're talking, depending on the organ, between 30 and 300 billion cells.
Okay, that's a lot.
Your typical, when you see a video of a scientist pipetting into a Petri dish, they're working with a couple of million. So the scaling is extremely difficult.
So a few million to several hundred billion.
And that's what stem cells do well. Because, exponential growth, it's who is it that had the quote, it's the most powerful, I think that was that an Einstein quote?
I think so,
there's compounding growth is the most powerful force in the universe. Exponential growth, it allows the stem cells to create billions of cells within a couple of weeks. And then we have to change the instructions from, okay, divide, to become heart cells or support cells for the tissue.
So this is, I think, a really important part of what you've been able to do recently in visiting your lab. Some of what you were showing me was essentially scaling up things that happen in many labs, growing enough cells, a few million, to be able to do single experiments, to the point where you can build an organ with hundreds of billions of cells. So maybe talk a little bit more about that scaling, because scaling isn't something we often do in academic labs. I think in industry that happens a lot, like somebody discovers a drug and industry works out how to make millions of those pills or billions of those injections. But in academic labs, we're not really built in the same way. So talk about how you've managed to really take on the scaling question.
Yeah, that's a really great question, and actually one that reviewers in our grants sometimes pick up on. Right? Is this academic? Is this industrial? Right? I think scaling is extremely important. I mean, when you look at impact that you can have, if you can make something cheaper and truly scalable, then that's a really good way to push our abilities forward. So in the same way as trying to manufacture big tissue, I'm just very attracted to the idea of scaling cell production. And it can be quite academic, actually. If you think about the cost of stem cell culture medium, right? It's about $600 a liter. Our reactors are 10 liters. That's $6,000 a liter per day. It's a lot of money for an academic lab. But what if we could engineer the cells to grow faster, or to not need the expensive ingredients in that media, or to differentiate not into one cell type, but actually form all of the cell types simultaneously.
Okay. Yeah.
There's really academic and synthetic biology ways that we're working on, and others, to make biology easier to work with and much more scalable.
Yeah.
So I think, there's definitely a role in academia to think about scale, and that's a really good way to amplify our impact. What it means physically is, instead of culturing these cells in a small media in a dish,
Right.
we have these up to 10-liter tanks of stem cells being cultured with a little impeller that stirs them all, and sensors to keep the oxygen levels happy, and media exchange where we're always adding fresh media and pulling out old medium.
Yeah.
And we have 18 of those reactors making the different cell types of the heart.
Yeah.
And that's really been manageable because of the production that we make our own media, we really optimize our own growth factor production, that we're able to handle that in-house makes that achievable.
Yeah, and I know we talked about this before, I'm sure you get asked this question a lot, but there has been this interest in lab-grown meat for eating, for nutrition, and some of that required, I guess, similar sorts of scaling.
Yes. So caveat: we grow human cells, so there's no lab-grown meat.
No lab-grown meat.
No lab-grown meat in
no hamburgers growing in the corner of the lab.
No.
Right.
But I'm from a very early point, I saw the synergy with the lab-grown meat industry, and there's actually those two, right? Scaling up to make meat quantities of cow cells, and scaling up to make organ quantities of human cells is very related requirements.
Right.
Now, I would argue that the lab-grown meat has a harder economic challenge, because they have to make a kilogram of cells and sell that for $2.
Right.
Whereas we have to make a kilogram of cells, and then the value, the life-saving potential of a heart, I would hope people would agree, is worth more than $2 for a life.
I think they would agree with that.
So we have an easier economic challenge,
Yeah.
but we have a much harder scientific challenge, because it doesn't just have to, it doesn't, it's not about the taste, it's about the function, right?
Yes,
so,
yes.
So it's
and your
very
tissue has to be alive.
And there's a few, we are in talks with some of these lab-grown meat companies. As you can imagine, they've really struggled with the economic argument. And some of them are starting to pivot towards, well, we've got all these facilities to make a lot of cells.
Right.
Human cells and tissues are much more valuable than a burger. So they're starting to pivot towards this space, which is very interesting and offers a lot of opportunities.
Yeah. Well, I really want to get to the heart, but before we get finally to that point, thinking about the lab-grown meat group, and other organs are presumably coming under the microscope for other scientists out there. Which ones have the most interest from the community, or the most interest from say, some of these pivoting lab-grown meat companies? Yeah,
there's probably the most attainable, what you might consider an organ that will be, I think, quite near-term successful, is the beta islet cells in the pancreas are looking very promising. So these are cells that are lost in diabetes type 1 or become dysfunctional in type 2 diabetics. And the reason why these are very attainable is there's a nice protocol to make these cells, but also you only need about a centimeter cube.
You do not need many,
Not need many, and they don't need to be placed in the pancreas to do their function. So you can inject them under the skin or somewhere where they can vascularize. Vertex is working on injecting them into the liver. And you basically add a little bit of pancreatic function in the liver, basically, that
like a micro pancreas
is looking really promising. The issue they have is those cells eventually either de-differentiate or die a lot of the time.
I see.
They fail to engraft, and a lot of that is because of the vasculature. So we're back to the same thing, right? If you can vascularize.
And presumably they choose the liver because it is a very vascular.
It's very vascularized and is able to kind of have a nice injection into I think they do a portal vein injection there. But if you can actually really structure the vasculature of that, you can improve potentially its engrafting success. So there are a couple of startups working in that space, and I think it's looking really promising.
That does sound really exciting. I mean, we talk in this forum here quite a lot about the potential for curative therapies, which is something over the last several decades that you would get laughed at for really talking about cures. It just wasn't a realistic aim. But there's another example potentially where if someone has type 1 diabetes, and you could provide them with a kind of micro pancreas that has produces insulin and that is sensitive to circulating factors, including, of course, glucose, you really could move someone to the point that they are effectively cured.
Yeah, yeah, and it's obviously a huge burden on society.
Yeah.
Right? There's a lot of diabetics, and so it's very promising.
So let's get to the heart. So
The heart of the matter.
The heart of the matter.
Someone had to.
There you are. Yeah, no, as a cardiologist, also someone who was attracted to the heart from my first physiology lab in Glasgow, so back in the UK as well, a little further north of Sheffield. I'm a pre-med student. It's like, I think, first year, and we have a lab, and everybody's excited because we're going to be able to play with a heart. And somewhere off behind the curtain, there's an animal donating its heart. And so we bring out a little dish, and there is a beating organ there. And our job is to hang it on a cannula, keep it alive for the next 6 hours. And I stared at that thing for hours, I kind of amazed at the sort of symphonic beauty of its contraction and coordination. And, you know, and pretty much in that moment, I'm like, I'm going to spend my life like learning everything I can about that organ. It isn't the simplest organ. I mean, your example that you just gave is like there are easier and lower hanging fruit to go grab, but you've taken on the heart. So tell me again why it was the heart that really drew you in, and let's see where you are. Like when can I transplant one of these into one of my patients?
Yeah, it's the story is quite straightforward, actually. Like how we stumbled upon things. Sometimes it's not the most exciting reason, but it's the reason.
Yeah.
And the reason is I was a postdoc in Jennifer Lewis's lab, myself and my friend Sebastian Uzel, who were trying to develop this way of vascularizing tissue. Had to choose a tissue, right? I mean, you know, you
pick one.
Pick one, right? Well, I mean, the way for us to pick it was we need a lot of cells for our method because we were working at really high cell densities. We needed billions of cells per experiment.
Right.
And we had 3 or 4 other students working with us on this project. And we thought, well, let's divide and conquer. So you do this tissue, you try and make those cells, you try and make those cells, and I'll try and make these cells. And whichever protocol works nicely in our hands and generates a lot of those cell types, we'll go with that.
That's where we're going.
And the answer was heart. I mean, it just turns out that to make heart from human stem cells, to make heart cells, is quite straightforward. It's a short protocol, uses only small molecules that are very cheap, so it's really scalable and it's really efficient. That became the tissue to test that on. And I think just like you, when I started making those cells and making tissues that you could just see beat and dance, I mean, come on, it's cool, right? I think engineers should all like love the heart. It's a really amazing organ.
It supplies all the other organs, as I constantly remind my fellow.
So that's kind of the honest origin story. But of course, heart disease, heart failure, right, is the number one cause of death, usually close with cancer. So the need is there, and we take our responsibility and goals very seriously with that. There's real people needing real help. And then on top of that, the structure is difficult. As you mentioned, it's a complex structure. Now, if you're going to build 3D printers that can make 3D shapes, the heart is a great way of showing that. Right? I can't imagine a way of casting a heart in the shape of a cube and having it work like a heart. Right? And it needs valves, it needs chambers, it has to be fluid dynamic. So the structure for the heart really matters. I'd argue more than any other organ. The structure of the macro scale structure. And sometimes I think of that as like if you were making a liver and it were cube shaped, so long as it had all the plumbing and microstructure of the little sinusoids and portal architecture, that it would probably work okay.
Probably
but
fine.
Macroscopically, if you looked at it on the table in the lab, it would look like a cube and it would probably work. That's not the case with the heart. So it's those combinations of big problem, the right tool for the right job, and just serendipity that way of making cells from human stem cells is the heart cells are quite easy to make.
Now the human heart has 4 chambers, of course, but across biology there are examples where there are 3. Frog is a good example. What is your approach to chambers and valves?
Yeah. So valves are, we'll start with the easy one, valves. We do have strategies to print valves out of a silicone that is looking pretty promising.
Yeah.
And we can integrate that silicone with the tissue that we're printing. So it's sort of a biohybrid design.
Yeah.
We still need to do long-term testing of these valves, right? Caveat, these are not fully tested yet.
But there are people with artificial valves.
But there are people doing great with artificial valves, right?
Yes.
It's not a solved problem, but you know, if I sort of joke with people in our lab that if we create a heart that works so well that it breaks its own valves, we're doing really well. We're doing really well. And we can always suture, we can always use one of these artificial valves into our tissue. So we do have a strategy for valves, but it's probably not mission critical.
Right.
In terms of chambers, we do make two separate cardiomyocyte types. We make ventricular and atrial cardiomyocytes. We don't make specific left ventricular and right ventricular. Those protocols are still not really fully developed. And secondly, you can actually use the right side of the heart as a left-sided heart. And doctors and surgeons do that. And while it does increase the risk of heart failure over a lifetime, it kind of works still.
Yes. So the opposite is probably a little better, a little easier, but yes, it is true.
So that way you can, still probably work with a ventricular cardiomyocyte. But we're ready when scientists create new, better ways to make the cells. We have all the machines and scaling ready to patch in. So we in terms of manufacturing the tissue, right? We load these cells into a printer.
Right.
We've got these new printers we're developing that print from many sides at once, so we can have these collaborating robotic printers that manufacture the different macrostructure of the heart. Once we have that in place, the tissue as we print it has a mayonnaise-like consistency. Right now, if you print mayonnaise on glass, right, in air, it will hold some shape, and then after a certain size, it's just going to flop over, right? So we work inside a support gel that holds what we print in place. And it just so happens that if you take heart cells and compact them in the centrifuge, jam them together, it makes a mayonnaise paste. So we can write this pasty material into the support gel, into the shape of a macrostructure, like a heart. And then the magic part is we come back into that printed tissue, and with finer print heads, we can start drawing vascular trees into that. So that mayonnaise-like consistency is liquid-like enough that we can write into it, but solid-like enough that whatever we print in stays in place.
Interesting.
And
so you print the muscle part first, and then you go back in
and write the fine detail, right? And we haven't done this yet, but also we hope to be able to write the conduction system right into the heart, right? These very specific high-resolution structures. And that way we solve some of the scaling problem. Because if you write everything with a really high resolution, it takes forever. It scales cubically. So I have my students do a little fun problem in my bioprinting class. If you want to print a liver with a 20 micron precision, single cell precision, it takes you 3,000 years. Right? It really
Long
scales
class.
badly. It's final in 6 weeks. They're not going to make that in time. So you want to kind of write some of the structure at low resolution, and then the parts that need to be high resolution, go in and write those separately. So that's
where does this self-organization part come in?
So partly in the microvasculature. As I mentioned, you put these blood vessel cells in the right environment and right condition, they will automatically form a vascular plexus, which is really helpful. Secondly, we need to somehow instruct those heart cells how to align. Right? If we have a heart, but every cell is facing in a different direction, then they're all going to be pulling against each other and not collaborating. So we have to instruct alignment.
This is about the motor control.
The motor, right. So that each heart cell, as it beats, it shortens, right? If this one shortens this way and that one shortens that way, the tissue is like, it doesn't know what to do. But if they're all aligned, then the tissue shortens, beats, generates a pressure. We have to instruct the cells how to align a little bit. And one way we can do that is to actually stress the cells in a 3D pattern that mimics the motion of the heart. most easy to understand is we build a little robot that moves the heart like the heart, and over time the cells start aligning and reproducing that alignment, so that when we remove that sleeve, they retain a bit of memory of how that
and they're under tension when that happens?
Under tension.
Okay.
So they align to the direction of maximum tension. So if you have a string and pull from both ends, the heart cells will align this way, right? But our heart isn't a string, it's a complex 3D shape, but actually it doesn't just beat like this. You may know it actually twists. It has a ringing effect, this helical alignment. And we found that if we twist the heart as it's developing, it actually retains that memory of the twists.
Yeah, I'm often teaching our students and residents and others about that twisting motion, because you can lay your hand on a patient's chest and feel it, right? And if the patient's heart is diseased, you'll feel that apex beat in a different way, because the twisting of the heart is a little different.
So, and it's important for its mechanical function. Otherwise, the outer parts of the heart wall start contracting up against the inner ones. They have to be in this sort of twisting architecture to really efficiently coordinate their beat across the thick wall.
The other part that I'm often emphasizing with students is that the heart also has to relax.
That's right.
And that actually, the suction created by the relaxation of the heart is also really important to bringing the blood in for the next beat.
Yeah, it's the difference between how soft the heart is in relaxation and how stiff it is in contraction, right? That dictates its cardiac output. And if you can't if it's not all about beating, it's also about relaxing. So yeah, I mean, these cells, this is a very important caveat, right? That no one's figured out how to make human stem cells differentiate into heart cells that beat with the strength of the adult cardiomyocytes. So that is a massive still hurdle that we or others are going to have to overcome. And so, both from a relaxation standpoint and a systolic contraction, we're not there yet with the basic biology. We know it works because we exist.
Yes.
It can be done.
Right.
But we need to be able to really mimic the right mechanical and chemical cues during development.
Do you think they're not mature enough, or
they're not mature enough?
I see.
But they're getting better. I mean, there's barely a week that goes by where another paper that makes better cells is coming out. And we're just trying to be ready with all of the other technologies needed to take those advances in the Petri dish right to some of this scale. So, we're very dependent on our other scientific colleagues and their hard work in making these cell types. But we also know, in part, if you don't have all the right structure, all the capillaries, all of the blood vessels, well-fed in a 3D environment where the forces are correct, you're not going to get full maturity, right? You can't rely just on chemicals alone.
So these two things actually meet each other in the middle.
Yeah.
You have a big grant from the ARPA-H group, which is all about big, bold ideas. That's where we started. I don't think anyone would dispute that you are following a big and bold idea. Where are you in terms of predicting when you might be at the point of having an actual organ?
So we will, through this grant, we will be in a pig model within 3 more years. It's
you're on track?
Be very honest.
3/4
of our milestones we're on track. A quarter of them are very tricky. But we are diverting resources to focus on that. The tricky parts are, as expected, getting capillaries working really nicely, robustly, keeping them open. That's really hard. Making the cells affordably at enough sort of scale and getting the repetition rate really frequent, so we get to practice a lot, is really hard. The macrophages, actually a really important cell type for development. They have a dual role. We usually think of them as part of the immune system in the adult, but actually they chaperone tissue development a lot during development as we're growing. So making these cells has been quite tricky for us to scale. So those are the regions where we're struggling. Where I think we're doing really well. We got a really cool printer that we're very excited about that can print very quickly. We have a code that can design the vasculature at a large scale for an organ that we're quite excited about. So there's big picture stuff that's going really well, and like every big project, parts that are really hard.
Well, you have come a very long way since you're dreaming about building things as a boy in Sheffield in the UK, and I was just really excited when you came to Stanford. I'm really excited when you came to our grand rounds here and shared the progress. And as a cardiologist and someone who sees patients with heart muscle disease, who are having kids with genetic heart muscle disease, I often talk to them about your work and the idea that progress is being made to the point where there might one day be a heart that could be made from your own cells, where you don't need that immune matching, you don't need somebody else's organ.
And we will be the first to acknowledge how hard that is,
Yeah.
but we shouldn't lose sight of that vision, because I think we have the lucky position in academia where we can try really hard things, because that's how you learn new things, right?
Yeah.
You try something bold and hard, and you learn what works better than expected and what needs more focus, and we hope to do that.
Thanks for joining us on The Future of Medicine.
Thank you.
Thanks, Mark.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.